On the floor of the deep ocean, in total darkness, at pressures that would crush a car engine, a whale skeleton lies on the sediment. It is not picked clean, it is not bare, it is covered, from one end to the other, in a living carpet of waving red plumes, thousands of them, moving in the slow current like breathing, like something enormous and patient and very much alive. They are not plants, not coral, not seagrass. They are worms, each one drilling into solid bone at this exact moment in water where no light has ever reached. And every single one of them has no mouth, no stomach, no digestive tract of any kind. There is no opening on their bodies through which food could enter, and yet the bone beneath them is disappearing, dissolving from the inside out, consumed by an animal that has no way of consuming anything. The worms visible on the bone are all female. The males exist, but they spend their entire lives inside the females, microscopic, never developing, never leaving, dozens to hundreds of them, living permanently inside each female, their only purpose to fertilize her eggs. These worms were doing this before the first whale ever existed. 100 million years ago, they fed on the bones of plesiosaurs and mosasaurs in seas that no longer exist. They survived the extinction that ended the age of dinosaurs. They have been here, in the darkness, doing this same thing ever since. Until recently, wherever a whale fell, they came. Every ocean, every depth, every skeleton. Then, at a monitored site off the coast of British Columbia, something changed. Whale bones were placed on the seafloor. Scientists waited. 10 years passed. The worms never came. Nobody knows why. And the silence where they should be is one of the most unsettling things happening in the deep ocean today. Take a moment to subscribe before we go deeper, and tell me in the comments, had you ever heard of these worms before tonight, or is this the first time? Let's go down. Far below the reach of waves and weather, the ocean becomes something entirely different. Beyond roughly 3,280 feet, the sea enters its largest realm, a vast landscape that spreads across about 65% of Earth's surface. It is the greatest habitat on the planet, yet much of it resembles a desert more than the crowded waters above. Distance alone separates these worlds, but that distance changes every rule by which life survives. Sunlight fades long before this depth is reached. By around 650 feet, the last useful rays have vanished, leaving no opportunity for photosynthesis. Without plants or algae producing fresh energy, every living thing below depends upon food created somewhere else. The deep ocean lives on leftovers, receiving only what slowly descends from the bright waters far overhead. That endless drift is known as marine snow. Tiny fragments of dead plankton, bits of organic tissue, waste from animals near the surface, and countless microscopic particles begin a gradual descent through the water column. Each piece becomes a meal before it falls much farther. Small creatures feed upon it, then larger ones feed upon them, until the original package of energy has been divided again and again during its journey downward. By the time that drifting material reaches around 13,000 feet, almost all of its original calories have already been consumed. Roughly 99% has disappeared into generations of hungry mouths suspended above. The remaining fragments settle onto the seafloor in quantities so slight that most residents survive by conserving energy rather than spending it. Growth slows, movement becomes economical. Encounters between animals may be separated by long stretches of time and remarkable distances. Against that sparse background, the arrival of a whale changes everything. A blue whale can weigh as much as 200 tons during its lifetime. When such an animal dies and its body descends into the darkness, it carries with it an astonishing reserve of stored energy. Researchers estimate that a single whale fall can deliver the caloric equivalent of roughly 2,000 years of ordinary marine snow to one location on the seafloor. Instead of a faint trickle spread across countless miles, an immense concentration of nutrients arrives all at once. The modern scientific study of whale falls began only in the early 1980s, making this one of the youngest fields in deep ocean biology. Since then, each discovery has revealed that a whale does not simply disappear into decay. Its body becomes the foundation of a living sequence that unfolds across decades, with every stage preparing conditions for the next. Large scavengers remove the richest tissues first. Smaller animals follow, harvesting what remains. Microorganisms continue the work long after flesh has vanished, gradually changing the chemistry surrounding the bones. Each community inherits a landscape reshaped by those that came before, adding another layer to a process that stretches far beyond the moment of death. Yet even after nearly everything visible has been consumed, the greatest reserve of nourishment still remains hidden from ordinary scavengers. Deep within the skeleton rests a store of fats and organic compounds, sealed inside hardened bone, protected beneath a mineral shell that few organisms can penetrate. Those hidden resources wait beneath the surface, preserved while countless creatures pass over them without access. It is there, within that final untouched storehouse, that the strange biology of Osedax finds its place. Among every participant in the long transformation of a whale fall, this worm alone is adapted to reach nourishment locked inside the skeleton itself, continuing a process that allows the deep ocean to draw life from what appears, at first glance, to be nothing more than bone. Long before any creature reaches the bones themselves, the whale becomes a signal carried by the water. As its immense body settles onto the seafloor, invisible compounds released by the earliest stages of decay begin spreading outward in broad currents. Across open stretches of deep ocean, animals equipped with remarkably sensitive chemical perception detect that drifting trail, following it across distances measured in miles until an isolated patch of seafloor becomes the center of sudden activity. The first arrivals are built for opportunity. Sleeper sharks emerge from the surrounding darkness, among the deepest diving sharks known to science. Six-gill sharks follow the same scent, accompanied by ray-tail fish that weave around the growing assembly. None of these animals discovered the whale by chance. Each intercepted a chemical message announcing that an enormous concentration of food had appeared where rich meals are exceedingly uncommon. Among the earliest visitors, hagfish begin one of the most unusual forms of feeding in the ocean. Rather than stripping flesh from the outside alone, they enter through natural openings or soft tissue, working their way deep into the carcass. Their bodies twist into tight knots, using their own muscles as anchors to generate greater pulling force. That remarkable movement allows them to tear loose pieces of flesh from within, gradually hollowing sections of the whale while remaining almost entirely enclosed by the body itself. Sleeper sharks contribute through persistence as much as strength. A single individual can consume as much as 44 pounds of tissue during one feeding session, then return again as long as nourishment remains. Every visit removes another portion of muscle or blubber, gradually exposing structures that had supported one of the largest animals ever to live. Countless smaller scavengers gather between those larger arrivals, each taking fragments too small to notice alone, yet substantial when combined over weeks and months. The pace of this transformation depends upon the size of the whale and the surrounding water conditions. A gray whale weighing about 40 tons may lose roughly 132 pounds of soft tissue every day during this period. Larger whales continue feeding entire communities for much longer, while colder environments often slow decomposition enough to extend the process. Across different oceans, the mobile scavenger stage may last as little as 4 months or continue for 2 full years before its work is largely complete. By then, nearly 90% of the whale's total wet weight has vanished. Muscles, organs, connective tissue, and thick layers of blubber have passed through hundreds of different mouths. What once appeared as a complete animal has become a broad framework of vertebrae, ribs, and skull resting upon the seafloor. From a distance, the skeleton may appear stripped clean, almost emptied of value. That appearance is deeply misleading. Hidden inside every large bone lies an extraordinary reserve of fats and other energy-rich compounds accumulated throughout the whale's lifetime. Unlike flesh, those resources remain protected beneath a hard mineral structure designed to support immense weight and life. Teeth cannot grind through it efficiently. Jaws cannot unlock what is sealed inside. Even the most capable scavengers gathered around the skeleton possess no biological machinery capable of extracting those hidden reserves. Their work, despite its intensity, reaches a natural boundary. The skeleton remains intact because conventional feeding ends where mineralized bone begins. Sharks move on in search of another opportunity. Hagfish abandon passages that no longer yield nourishment. Rat-tail fish disperse into the surrounding depths, leaving behind a structure that still contains remarkable wealth beyond their reach. What rests upon the seafloor at this stage is neither empty nor finished. It has become something entirely different, a reserve of nourishment enclosed within stone-like walls, awaiting an organism whose relationship with bone bears almost no resemblance to the feeding strategies of any scavenger that came before. The first sign of the next community is surprisingly delicate. From the surface of the exposed skeleton rise slender crimson plumes, each belonging to a female Osedax anchored firmly within the bone below. Most measure between 1 and 3 inches long, their bodies extending upward from the skeleton like tiny stems. At the tip of each retractable trunk rests a crown of 4 feathery palps that drift with passing water, gathering dissolved oxygen that sustains the worm and the unseen partnership living inside it. Everything beneath those plumes defies ordinary animal anatomy. An Osedax female has no mouth through which food can enter. It has no anus through which waste can leave. Between those absent openings lies no digestive tract at all. The familiar pathway followed by nearly every other animal has disappeared entirely. For years after its discovery, biologists struggled to understand how a creature lacking every conventional feeding organ could thrive while rooted inside solid bone. The answer begins where the lower end of the female presses against the skeleton. Instead of scraping, biting, or drilling with hardened structures, the worm relies upon chemistry. Specialized tissue at the base of its body releases acid directly onto the mineral surface of the bone. Contact alone begins dissolving the hard outer layer, gradually softening material that resisted every scavenger before it. Rather than forcing its way inward, the worm patiently opens a passage by changing the bone itself. As that mineral barrier weakens, an intricate system of root-like filaments spreads through the interior. These living branches extend into newly opened spaces, dividing again and again until they occupy a widening network beneath the surface. From above, only the red plumes remain visible, yet below them, an elaborate structure is steadily expanding through chambers that were once solid bone. Neighboring females often extend their own branching tissues nearby, creating overlapping networks that remain hidden from every passing animal. Those root filaments hold the true engine of the entire process. Living permanently inside the worm's own tissues are colonies of endosymbiotic bacteria, microscopic partners that have become inseparable from their host. They are not temporary visitors carried in from the surrounding water. They spend their lives enclosed within the worm, receiving shelter while performing the chemical work the animal cannot accomplish alone. Once acid has released collagen and energy-rich lipids from the surrounding bone, the bacteria begin breaking those compounds apart. Their metabolism transforms complex biological material into simpler nutrients capable of passing directly through the surrounding tissues. The worm never digests its meal in the familiar sense. Instead, it absorbs the products created by its bacterial partners, drawing nourishment across its own cells without ever possessing a stomach or intestine. This remarkable arrangement explains why the Latin name Osedax, often translated as bone devourer, captures only part of the story. The worm itself does not consume bone in the direct manner suggested by that name. It dissolves the mineral structure, exposing hidden organic reserves. The bacteria enclosed within its roots process those newly available compounds, and the worm receives the resulting nourishment. 3 participants, bone, bacteria, and worm, become parts of one continuous biological system. Researchers eventually recognized another remarkable parallel within this chemistry. The acid released from Osedax root tissue resembles the process used by specialized mammalian bone cells that continually reshape living skeletons throughout life. Those cells dissolve old bone so new bone can replace it. Osedax arrived at a strikingly similar chemical solution through an entirely separate evolutionary path, demonstrating how distant branches of life sometimes discover comparable answers to very different challenges. As colonies mature, the hidden architecture beneath the surface grows increasingly elaborate. A single female may extend her roots so deeply that neighboring channels intersect beneath the bone, forming hollow passageways invisible from above. One whale vertebra can support thousands of females at once, each operating its own network of acid, releasing tissue and bacterial partners. Together, they transform what once seemed an impenetrable skeleton into a living landscape shaped from within, one microscopic passage at a time. From above, the colony appears almost uncomplicated. Hundreds or even thousands of crimson plumes sway over the whale's bones, each marking the presence of a thriving worm beneath the surface. Every visible individual belongs to the same sex. Every plume, every branching root system, every acid dissolving its way through bone is produced by a female. The males exist as well, yet no remotely operated vehicle has ever captured them moving across a whale skeleton, because they spend their entire lives concealed within the bodies of the females themselves. In most known species of Osedax, a male never grows into the form that might ordinarily be expected of an adult worm. Instead, it remains microscopic, preserving the simplified anatomy of a newly settled larva throughout its entire existence. Development pauses at an extraordinarily early stage. Rather than building a larger body capable of feeding independently, the male retains only those structures necessary to survive long enough to fulfill a single biological role. Its own digestive system never develops. There is no mouth to gather food and no intestine to process it. At the beginning of life, the tiny male depends entirely upon the yolk supplied within the egg from which it hatched. That inherited reserve provides enough nourishment for early survival, but it cannot sustain an independent future. The solution lies not in finding food, but in finding a female. Surrounding the upper portion of every female is a soft, gelatinous tube, a translucent sleeve extending between the waving plumes and the surface of the bone. What appears from the outside to be little more than a protective covering serves another purpose altogether. It becomes a permanent refuge for the males, enclosing them within a sheltered space where they remain for the rest of their remarkably abbreviated lives. An average female commonly hosts somewhere between 50 and 100 males inside that tube. Older and larger females may support far greater numbers. Researchers have documented harems exceeding 600 microscopic males living together around a single female, all occupying the same narrow enclosure while continuously producing sperm. No other responsibility shapes their existence. They do not leave, forage, compete for territory, or construct burrows. Their entire contribution to the colony is reproductive. As the female continues extending her roots deeper into the skeleton, she does not simply retain the males acquired during her earliest days. New larvae drifting through the surrounding water are capable of detecting chemical signals released by an established female. Instead of settling onto exposed bone, some enter her gelatinous tube and remain there, joining the existing population. Throughout the female's lifetime, this process allows new males to replace older individuals, maintaining an ongoing source of fertilization. Perhaps the most remarkable aspect of this arrangement is that no larva begins life genetically assigned to become either male or female. Every larva carries essentially the same developmental potential. The decision emerges only after it encounters its surroundings. A larva that lands directly upon an unoccupied whalebone begins transforming into a full-sized female, extending roots into the skeleton and establishing a colony of its own. Another larva possessing the very same genes may instead encounter a mature female first. Entering her tube redirects its entire development, producing a dwarf male rather than another bone-dissolving female. Biologists describe this phenomenon as environmentally determined sex. The surrounding conditions, rather than inherited genetic instructions alone, guide the developmental pathway each larva follows. Location becomes destiny. Arrival upon bare bone leads toward one future, while arrival beside an established female leads toward another that is dramatically different, despite beginning with nearly identical biological instructions. Once fertilization has taken place, the female releases eggs through an oviduct into the surrounding water. Ocean currents then carry those developing larvae across the deep sea, where each drifting life begins the same search anew. Some eventually encounter untouched bone and become founders of entirely new colonies. Others find an already flourishing female and disappear within her translucent shelter, remaining almost invisible for the rest of their lives while sustaining one of the most unusual reproductive systems known anywhere in the animal kingdom. Scientific understanding often advances by recognizing patterns. Once enough observations accumulate, those patterns begin to resemble rules. By the year 2014, one rule appeared especially secure within the study of Osedax. Every known species followed the same remarkable arrangement. Large females occupied the bones, while microscopic males spent their entire lives sheltered inside them. Across oceans and across species, the pattern seemed so consistent that it appeared woven into the identity of the entire genus. That certainty began to shift when researchers from the Scripps Institution of Oceanography, the Monterey Bay Aquarium Research Institute, and the Western Australian Museum examined specimens collected from nearly 2,300 feet beneath the waters off the California and Oregon coast. At first glance, the worms belonged unmistakably to Osedax. Their feeding structures matched the familiar design, and their relationship with bone followed the same general plan. Yet one feature refused to fit anything scientists believed they understood. The males were no longer microscopic. Instead of remaining frozen in an immature larval form, these males had developed into adults, approaching the size of the females themselves. Compared with the dwarf males known from every previously studied species, they were tens of thousands of times larger. They possessed complete bodies, capable of independent life, abandoning a developmental pathway that had seemed fixed across the entire evolutionary history of the group. The new species received the name Osedax priapus, honoring the ancient Greek god associated with male fertility. The choice reflected an especially unusual feature of the males. Their bodies could stretch to roughly 10 times their resting length, allowing them to physically reach nearby females during reproduction. This was a completely different strategy from the concealed harems found in other members of the genus, where fertilization occurred from within the female's gelatinous tube rather than through direct contact between similarly sized adults. These males also shared another ability long thought unnecessary. They fed independently on bone. Like the females, they established themselves upon the skeleton and obtained nourishment through their own association with bone processing. The tiny males found in other Osedax species never perform this task because their entire existence depends upon living inside a female. In Osedax priapus, both sexes became active participants in feeding as well as reproduction. The biological surprise extended far beyond differences in appearance. Evolutionary theory suggested that the genetic instructions needed to build a full-sized adult male should have faded long ago. When genes remain unused across millions of years, mutations gradually accumulate. Without natural selection preserving their function, those sequences often deteriorate until they can no longer produce the structures they once encoded. After such an immense span of evolutionary time, the expectation was straightforward. The blueprint for a fully developed male body should no longer exist, yet it remained intact. Hidden within the lineage all along were functional genetic instructions, capable of producing an adult male whenever evolutionary conditions favored that outcome. Rather than inventing an entirely new body plan, the species revealed an ancient developmental possibility that had endured through ages during which it appeared unnecessary. The change represented not a fresh invention, but the return of a capability believed to have been lost. Lead researcher Greg Rouse described the discovery as the first known example in the animal kingdom of such a dramatic reversal from dwarf males. Biologists refer to this type of return as an atavism, a reappearance of ancestral traits after generations in which they seemed absent. Co-author Robert Vryenhoek emphasized the same point, noting that the genes responsible for constructing full-sized males should have broken down through prolonged disuse, yet they had somehow remained complete and functional. Despite this extraordinary discovery, the broader story of Osedax changed very little. Osedax priapus stands alone as the only known species to abandon the familiar arrangement. Every other confirmed member of the genus continues the older pattern, with hundreds of microscopic males living within each female, preserving a reproductive system that has endured across countless generations beneath the ocean floor. Scientific history is filled with discoveries that arrived while someone was searching for something entirely different. The story of Osedax began in exactly that way, on the 6th of February, 2002, when marine biologist Robert Vryenhoek stood aboard the research vessel Western Flyer as a remotely operated vehicle explored the floor of Barkley Canyon off the coast of California. The expedition was following a large sonar return that researchers believed marked an extensive clam bed resting nearly 9,500 feet below the surface. Instead, the cameras revealed a gray whale skeleton. Even that was not the greatest surprise. Stretching across the bones from one end to the other was what looked at first like a thick crimson carpet. It coated vertebrae, ribs, and skull so completely that the underlying skeleton seemed wrapped beneath living fabric. Then the image changed in a subtle but unmistakable way. The crimson covering shifted with the surrounding water. Countless slender plumes moved together, revealing that the carpet itself was alive. Vregenhoek and his colleagues watched the video feed from the control room without recognizing what they were seeing. Nothing in the scientific literature matched the organisms covering the whale. They resembled neither coral nor familiar tube worms, and their association with exposed bone made them even more puzzling. The expedition had arrived expecting to investigate one type of deep-sea habitat, only to encounter an entirely unknown form of life. Specimens were collected and brought to the surface for closer examination. Within 48 hours, genetic analysis provided the first important clue. The mysterious worms belonged within the broader group of deep-sea tube worms associated with hydrothermal vents, yet they fit no described genus. Their ancestry was recognizable, but their identity was entirely new. Evolution had produced a branch of this family, unlike anything previously recorded. Recognizing the importance of the find, the research team contacted Greg Rouse, then serving as curator at the South Australian Museum. After examining the material, Rouse reached the same conclusion. The worms represented a completely undescribed genus. Science had no existing name for them because no one had documented them before. A lineage that had almost certainly existed for millions of years entered the scientific record only because a remotely operated vehicle happened to investigate the wrong sonar target. The first descriptions identified two distinct species inhabiting the same whale skeleton. They received the names Osedax ruboplumis and Osedax frankpresi. The genus itself was named Osedax, derived from Latin words meaning bone devourer. That name was chosen only 2 days after the initial discovery, long before researchers understood the extraordinary chemistry by which the worms actually obtained nourishment. They recognized what the animals accomplished before they understood how it happened. When scientists returned to the same whale only 2 years later, another unexpected pattern had emerged. Osedax ruboplumis, once abundant across the skeleton, had been replaced almost entirely by Osedax frankpresi. One species appeared to give way to another as the whale fall aged, suggesting that even among bone-eating worms, ecological succession shaped life upon the same carcass. The skeleton had become not only a habitat, but a changing community whose occupants shifted through time. As additional whale falls were investigated around the world after 2004, the known diversity of the genus expanded steadily. Researchers eventually confirmed 32 species inhabiting waters ranging from only about 32 feet below the surface to depths exceeding 13,000 feet. Each discovery demonstrated that the original finding in Barkley Canyon had not uncovered an isolated curiosity, but revealed a globally distributed lineage hidden in plain sight beneath the oceans. The earliest years following the discovery carried one final mystery. Despite careful examination of collected specimens, researchers could not locate any males at all. For a time, some wondered whether these worms might reproduce without them. Only later did the answer emerge, concealed within the females from the very beginning, waiting to overturn yet another assumption about one of the ocean's most extraordinary animals. A whale's skeleton does not remain the same after Osedax arrives. Beneath the graceful plumes visible above the surface, the bone begins changing into something with a completely different purpose. What once served as the framework of an enormous animal gradually becomes architecture for an entire community. The transformation is not accidental. Every tunnel dissolved into the skeleton alters the landscape that countless other organisms will eventually inherit. As each female extends her branching roots through the mineral structure, she leaves behind far more than narrow passageways. The expanding network produces chambers, cavities, and interconnected channels throughout the interior of the bone. From the outside, a vertebra may still appear largely unchanged, yet hidden beneath its surface is an increasingly intricate framework carved by thousands of separate worms working at the same time. Bone that was once dense and comparatively uniform slowly develops the complexity of a natural reef enclosed within itself. Computed tomography scanning has allowed scientists to look inside these transformed bones without destroying them. The resulting images reveal a remarkable lattice of merged root channels spreading through the interior like the branching veins of a leaf. Individual tunnels intersect with neighboring ones until the original structure becomes almost unrecognizable. As colony density increases, the internal architecture grows progressively more elaborate, creating countless protected spaces that did not exist when the whale first reached the seafloor. Those newly formed spaces soon become homes. Tiny invertebrates settle inside narrow cavities, microorganisms coat exposed surfaces, and species unable to establish themselves upon smooth bone discover shelter within the expanding maze. Research comparing whale falls colonized by Osedax with those lacking the worms consistently shows much greater biological diversity where these hidden networks have formed. The worms are not simply extracting nourishment for themselves. By reshaping the physical structure of the skeleton, they generate habitat that many other organisms depend upon for survival. Their influence extends beyond architecture into chemistry. As bacterial partners inside Osedax process collagen and energy-rich lipids released from the dissolved bone, decomposition continues through a series of microbial reactions. Among the products generated during this extended breakdown is hydrogen sulfide, a compound that gradually diffuses into the surrounding sediment and nearby water. The appearance of hydrogen sulfide signals that the whale fall has entered an entirely different stage of its existence. This next community is known as the sulfophilic stage, meaning a community that thrives through an affinity for sulfide. Unlike the ecosystems near the ocean surface, where food originates from sunlight captured by photosynthesis, this one depends upon chemosynthesis. Specialized microorganisms obtain energy directly from chemical reactions involving hydrogen sulfide, allowing them to manufacture organic material without any reliance upon light. Those microorganisms become the foundation of an entirely new food web, supporting animals that flourish where ordinary sources of nourishment are absent. The duration of this stage is remarkable. Depending upon the size of the whale and the amount of energy remaining inside its skeleton, the sulfophilic community may persist for anywhere between 15 years and well over 50 years. Long after flesh has disappeared and the earliest scavengers have departed, the bones continue supporting a thriving ecosystem whose existence depends upon chemical energy released from within the skeleton itself. Without Osedax, that sequence may never fully unfold. Organic compounds remain trapped inside mineralized bone, hydrogen sulfide production is greatly reduced, and the chain of biological events leading to a mature sulfophilic community weakens before it has the opportunity to develop. One of the richest habitats associated with a whale fall may never assemble because the physical and chemical groundwork was never completed. For that reason, Ecologists describe Osedax with a title far more revealing than scavenger. They recognize it as an ecosystem engineer, an organism whose daily activities reshape the surrounding environment in ways that benefit many other species. Every passage dissolved into bone, every microscopic partnership maintained within its roots, and every chemical change released into the seafloor contributes to a habitat far larger than the worm itself. Through the work of countless individuals hidden inside the skeleton, a dead whale becomes the foundation for decades of continuing life. There are places on the ocean floor where life flourishes without ever depending upon the Sunday. One of the best-known examples is a hydrothermal vent, a crack in the seafloor where superheated water rich in dissolved chemicals rises from deep within the earth. Around these openings, entire ecosystems develop without photosynthesis. Instead of drawing energy from light, microscopic organisms harvest energy directly from chemical reactions, supporting dense communities in places where conventional food webs could never exist. Something remarkably similar unfolds around an aging whale skeleton, although on a much smaller scale and through a very different source of chemistry. The whale does not release heat from the Earth's interior. It releases opportunity stored within its own bones. As Osedax and its bacterial partners continue processing collagen and energy-rich lipids, hydrogen sulfide gradually spreads into the surrounding sediment and nearby water. That invisible compound becomes the foundation of another ecosystem, one powered by chemistry rather than sunlight. Chemosynthetic bacteria are the first to benefit from this changing environment. Instead of relying upon organic particles drifting down from distant surface waters, they obtain energy directly from reactions involving hydrogen sulfide. Using that energy, they manufacture new organic material that becomes nourishment for other forms of life. In this way, the whale skeleton begins supporting a food web fundamentally different from the one that existed during the earlier stages of decomposition. As these bacterial communities expand, familiar residents of other chemically powered habitats begin appearing among the bones. Certain lineages of tube worms settle into the growing ecosystem alongside particular bivalves and carefully adapted gastropods. These animals are not strangers to environments rich in hydrogen sulfide. Many are also found around hydrothermal vents, where they have evolved to flourish in conditions unsuitable for most marine life. The whale fall offers them another island of chemistry within the immense expanse of the deep ocean. Although much smaller than a hydrothermal vent field, the sulfophilic stage reproduces many of the same ecological relationships. Microorganisms transform chemical energy into living tissue. Grazers and filter feeders depend upon those microorganisms. Predators then feed upon the growing community above them. Layer by layer, a complex web of interactions develops from a source of energy hidden inside what once appeared to be an empty skeleton. The endurance of this community is equally remarkable. Depending upon the size of the whale and the remaining stores of bone lipids, the sulfophilic stage can continue for at least 15 years and often well beyond 50 years. During that span, the whale skeleton remains an active center of biological productivity long after every trace of flesh has disappeared. Time affects this ecosystem differently than the rapid feeding frenzy that first greeted the whale's arrival. Growth becomes measured in years rather than weeks, yet the community continues expanding through steady chemical exchange. Researchers witnessed that persistence directly when they revisited a whale fall discovered off the coast of British Columbia in 2009. Returning during the year 2024, they found the skeleton resting about 4,100 feet below the surface. still supporting an organized and thriving community after 15 full years. The whale had become far more than the remains of a single animal. It had matured into a stable ecosystem sustained by processes unfolding deep inside its bones. The largest whales possess even greater potential. A blue whale stores such extraordinary quantities of lipids within its skeleton that researchers estimate a single carcass may contain enough chemical energy to sustain this type of chemosynthetic community for as long as a century. Few habitats in the deep ocean remain productive for such extended periods while occupying a single fixed location. Among every stage in the life of a whale fall, the sulfophilic phase supports the greatest diversity of organisms. Its abundance depends upon work completed much earlier, when Osedax dissolved mineral barriers and unlocked the hidden reserves sealed inside the skeleton. By reshaping the bones from within, the worms prepare the conditions that allow one of the richest communities in the deep sea to emerge, linking the fate of countless species to a process that begins with a single colony hidden beneath crimson plumes. Counting life around a whale fall reveals a story that no single photograph can capture. A camera may record worms, clams, snails, crustaceans, and fish gathered around the skeleton. Yet every careful survey uncovers another layer of inhabitants occupying different surfaces, different sediments, and different moments in the long progression of decay. What first appears to be one isolated carcass gradually resolves into one of the richest biological communities ever documented on the deep seafloor. Researchers have now recorded more than 400 species associated with whale falls across the world's oceans. They belong to numerous branches of the animal kingdom, representing a remarkable variety of body plans, feeding strategies, and evolutionary histories. Some arrive only briefly before moving on. Others remain for years. Still others complete their entire lives within the changing habitat created by a single whale skeleton resting on the ocean floor. Among those, many inhabitants are an especially remarkable group. At least 21 confirmed species are known only from whale falls. They have never been found living successfully in any other environment. Their existence is tied completely to this rare sequence of ecological events. If no whale reaches the seafloor, these specialists possess no alternative habitat waiting elsewhere. Their entire evolutionary story has unfolded around the appearance, transformation, and gradual disappearance of whale skeletons. Those 21 specialists do not belong to one closely related family. Instead, they are distributed across 5 separate biological phyla, representing lineages that diverged from one another hundreds of millions of years ago. This is not merely a collection of similar species adapting in parallel. It is deep evolutionary diversity expressed through fundamentally different forms of life, all converging upon the same extraordinary habitat through entirely separate histories. Such specialization requires immense stretches of time. Scientists believe whale falls have persisted long enough for natural selection to shape organisms that depend upon them completely. Generation after generation encountered the same predictable sequence of changing conditions until entire species evolved around opportunities found nowhere else. The habitat itself became stable enough across geological time to guide evolution toward increasing specialization rather than general survival. Even within a single whale fall, diversity is unevenly distributed. Early scavenging stages support many animals, but those communities are temporary by nature, following flesh that eventually disappears. The greatest richness develops later, during the sulfophilic stage initiated through the work of Osedax. as hydrogen sulfide sustains chemosynthetic microorganisms. An increasingly complex food web expands around the skeleton, supporting the greatest concentration of specialized organisms documented during the whale fall's long existence. This pattern reveals that Osedax contributes far more than its own presence. The worm prepares conditions, allowing hundreds of additional species to occupy the habitat. If its role is removed from the sequence, the consequences spread backward through the ecological chain. Bone lipids remain largely inaccessible. Hydrogen sulfide production declines. The chemosynthetic community develops only weakly or fails to establish itself altogether. The extraordinary diversity associated with the mature whale fall never reaches its full expression because its foundation was never completed. The effects become even more profound for the true specialists. Species that evolved exclusively for whale falls cannot simply relocate to neighboring reefs, hydrothermal vents, or ordinary stretches of seafloor. Their anatomy, behavior, and life cycles have been shaped around a habitat unlike any other. Generations of adaptation have narrowed their ecological possibilities until the whale fall itself became their only dependable home. Viewed in this light, the importance of Osedax extends well beyond the fate of one unusual worm. Its patient work inside the skeleton influences whether an ecosystem reaches its full biological potential or remains an incomplete version of itself. More than 400 documented species benefit from that transformation, while 21 known specialists depend upon it entirely. Beneath the ocean, where every opportunity for life is hard-won, the future of an entire community can rest upon an animal only a few inches long, working unseen within the bones of a whale. An adult Osedax will never leave the bone where its life began. Once its roots spread into the skeleton, every possibility of movement ends. The worm remains anchored for the rest of its existence, growing, reproducing, and gradually reshaping the bone beneath it, without traveling even a few inches. If the species is to reach another whale, it must do so before adulthood ever begins. That journey belongs entirely to the larva. Released into the surrounding water after fertilization, each larva enters the immense circulation of the deep ocean. It possesses no map, no destination, and no guarantee that another whale skeleton lies ahead. Ocean currents become its only means of travel, carrying it across distances that may span hundreds of miles before the opportunity to settle finally appears. Most larvae, almost certainly, never encounter suitable bone at all. The few that do become the founders of entirely new colonies. To marine ecologists, each whale fall resembles an island rising from an otherwise sparse landscape. Vast stretches of the abyss contain little concentrated nourishment, yet scattered across that enormous plain are occasional centers where life flourishes for decades. Every whale skeleton becomes an isolated refuge separated from the next by miles of comparatively unproductive seafloor. Although the islands never break the ocean's surface, they function in much the same ecological way. This idea forms the basis of what scientists call the stepping stone hypothesis. Rather than existing as disconnected populations, Osedax and many other whale fall organisms maintain healthy numbers by dispersing from one whale fall to the next. Each skeleton serves as both a home for the present generation and a launching point for the next. Larvae departing one productive habitat eventually discover another, preserving genetic exchange across enormous regions of the ocean through an uninterrupted chain of temporary refuges. The importance of those stepping stones reaches far beyond Osedax itself. Many species associated with whale falls also appear around hydrothermal vents and cold seeps, environments that likewise depend upon chemical energy rather than sunlight. The overlap is substantial enough to suggest that these habitats have remained connected through evolutionary time. Whale falls may provide intermediate refuges where populations survive between larger, chemically powered ecosystems, allowing organisms to expand their distribution across entire ocean basins without requiring continuous habitat. In this way, the skeleton of a whale becomes more than a local event. It forms part of a continental scale network stretching across the deep sea. Every productive whale fall strengthens the connections linking distant populations. Every successful colony produces larvae capable of continuing the chain, carrying genes and species from one isolated habitat toward another that may lie far beyond the horizon of any underwater camera. The placement of these biological islands is not random. Whale falls appear only where whales live, migrate, feed, and ultimately die. Ocean currents determine where carcasses drift after death, while whale populations determine how frequently new skeletons reach the seafloor. Together, those factors establish the spacing of the stepping stone network. The geography of the deep sea is shaped not only by mountains and trenches, but also by the movements of living whales in the waters far above. As whale populations increase, more skeletons eventually descend to the abyss, creating additional habitats and shortening the distance between them. Larvae encounter suitable settlement sites more often, improving the chances that populations remain connected across wide regions. When whale numbers decline, the opposite pattern emerges. New whale falls become less frequent. The spaces between productive habitats widen, and successful dispersal becomes increasingly uncertain. The health of this invisible network, therefore, depends directly upon the abundance of the animals swimming near the ocean surface. Every living whale represents not only a participant in today's marine ecosystem, but also a future stepping stone capable of sustaining life on the seafloor for decades after its own journey ends. Through those scattered islands of bone, organisms only a few inches long remain connected across some of the largest and least explored landscapes on Earth. Long before anyone lowered cameras into the abyss, the future of countless deep-sea communities had already been altered at the ocean's surface. The connections between distant whale falls depended upon living whales completing migrations that had unfolded for millions of years. Every generation carried the possibility of another skeleton descending into the depths, another island appearing across the vast plain, another opportunity for drifting larvae to continue an ancient chain stretching from one ocean basin to another. Before industrial hunting transformed the seas, global whale populations are estimated to have numbered in the tens of millions across many different species. Blue whales, fin whales, sperm whales, humpbacks, right whales, and many others crossed enormous migratory routes that repeatedly linked feeding grounds and breeding grounds. Over time, those migrations produced a steady rhythm of whale falls scattered across the seafloor. No single skeleton lasted forever, yet together they formed a continually renewing network of habitats separated by distances that wandering larvae could still overcome. That pattern changed with remarkable speed during the late 19th century and continued through much of the 20th century. Expanding industrial whaling fleets operated across every major ocean, pursuing the largest whales with unprecedented efficiency. Steam-powered vessels, explosive harpoons, and factory ships transformed animals that had once been difficult to capture into commercial resources harvested on a global scale. The consequences reached far beyond the waters where the hunts occurred. Among the most heavily affected was the blue whale. In the Southern Ocean, Its population eventually declined to an estimated 1 to 3% of its pre-industrial abundance before international protections began reversing the trend. Other great whales experienced similarly severe reductions. The North Atlantic right whale approached the edge of disappearance, and even today, fewer than 400 individuals remain. Many surviving populations became only small fragments of what had once inhabited the oceans. Each whale removed from the sea represented more than the loss of a living animal. It also represented a whale fall that would never descend through the water column. An entire future ecosystem disappeared before it had the opportunity to exist. Bones that might have nourished Osedax, sustained chemosynthetic bacteria, and supported generations of specialized organisms never reached the abyssal plane. The missing skeletons accumulated year after year until vast stretches of the deep ocean contained fewer biological islands than they had for countless ages. The stepping stone network responded accordingly. Greater distances separated productive habitats, making successful dispersal increasingly uncertain for larvae drifting through deep currents. Populations that had once exchanged genes across broad regions became more isolated from one another. As reproductive connections weakened, genetic diversity likely declined within many species dependent upon recurring whale falls. The invisible architecture supporting these communities became fragmented, even though the changes remained hidden beneath thousands of feet of water. Scientists suspect that some whale fall specialists may have disappeared from particular regions during this era. Others may have vanished entirely before anyone realized they existed. The uncertainty remains because systematic exploration of the deep sea had scarcely begun, while industrial whaling reached its greatest intensity. Entire communities could have contracted or disappeared without leaving any historical record beyond the absence now impossible to measure. International restrictions introduced during the 1980s gradually reduced commercial whaling, allowing several whale populations to begin recovering after generations of decline. Progress has been uneven, and some species continue to struggle, yet increasing whale numbers also mean increasing opportunities for future whale falls. As more great whales complete natural lives, additional skeletons once again descend toward the seafloor, slowly restoring connections between distant habitats that had long remained separated. Recovery beneath the ocean unfolds on a timescale measured in decades rather than seasons. Every new whale fall begins rebuilding pathways interrupted long before the first Osedax colony was ever observed by science. The ancient network has started weaving itself together once more, thread by thread, across the darkness of the deep ocean, even as another challenge emerged to shape its future. The story seemed complete for a time. As researchers traced the history of Osedax through genetic comparisons with its closest tube worm relatives, the evidence appeared to point toward an elegant partnership between mammals and worms. Early molecular dating suggested that the lineage diverged roughly 45 million years ago, a period that closely matched the rise of large archaic whales during the Eocene. The explanation fit comfortably within the broader history of evolution. Whales had become enormous. Their skeletons created an untapped source of nutrition on the deep sea floor, and Osedax had seemingly evolved to exploit that remarkable opportunity. For years, this interpretation remained persuasive because it united genetics, ecology, and the fossil record into a single narrative. Whale bones appeared to have created a new ecological niche, while the strange anatomy of Osedax looked perfectly suited to occupy it. The apparent timing reinforced the idea that one innovation had naturally followed another. Yet the fossil record has a habit of preserving details that reshape even the most convincing scientific stories. That transformation began in 2015, when researchers at Plymouth University looked not toward ancient whales, but toward creatures that had disappeared tens of millions of years earlier. Dr. Nicholas Higgs and Dr. Sylvia Denise examined fossilized marine reptile bones dating back roughly 100 million years. Their material included plesiosaur specimens recovered near Cambridge in England and fossil sea turtle remains from Burham in England. Rather than relying only upon the exterior of the fossils, they employed computed tomography scanning to peer through the stone without damaging it. Inside those ancient bones, the scans revealed something astonishing. Networks of narrow tunnels branched outward beneath the fossil surface, forming radiating patterns strikingly similar to those produced by modern Osedax. The borings did not resemble disease, fractures, blood vessel architecture, or any other known biological process preserved within fossil bone. Instead, their shape matched the intricate root systems created by living bone-eating worms as they dissolve mineralized tissue and spread through its interior. The implication reached far beyond the fossils themselves. If those tunnels had indeed been produced by Osedax, then the worms had already been consuming the bones of plesiosaurs roughly 100 million years ago. That date preceded the appearance of the earliest whales by approximately 55 million years. The elegant narrative that had linked the origin of Osedax to whale evolution could no longer stand unchanged. A different picture gradually emerged. Rather than evolving because whales created a new opportunity, Osedax had already occupied that ecological role for tens of millions of years. Whales entered an ancient system instead of creating it. As giant marine reptiles disappeared near the close of the Cretaceous period, enormous whale skeletons eventually became the next abundant source of bone reaching the deep seafloor. The worm did not need to reinvent its biology. It merely continued performing the same remarkable work upon different giants, separated by immense spans of geological time. Evidence supporting that revised history continued to accumulate. During 2024, researchers published additional computed tomography studies documenting Osedax-style borings in 3 more plesiosaur specimens. They also identified the first comparable structures within 2 mosasaur fossils recovered from Belgium and the southeastern United States. Those discoveries greatly expanded both the range of host animals and the geographic distribution preserved in the fossil record. The pattern now stretched across England, Belgium, and the United States, indicating that Osedax already possessed a broad Atlantic distribution during the Campanian Age, between roughly 83 million and 72 million years ago. By 2025, further examination of Cretaceous marine reptile collections in the United Kingdom revealed 5 distinct boring morphotypes preserved within fossil bone. Those differing patterns suggested that multiple Osedax species already existed, while marine reptiles still ruled the ancient seas, long before the first whale ever began its descent into the deep. Not every skeleton has an equal chance of entering the history preserved in stone. Long after an animal's life has ended, another process determines whether its remains become part of the fossil record or disappear forever. Between death and discovery lies an interval shaped not only by geology, but by biology. During that interval, Osedax has influenced the story of ancient oceans for far longer than anyone once imagined. For a bone to become a fossil, it must remain physically intact long enough for burial beneath accumulating sediment. Protected from continued destruction, Minerals gradually replace the original organic material while preserving much of the bone's structure. Over immense spans of time, that buried skeleton may emerge as evidence carried forward across millions of years. The process depends upon stability. Bones that are broken apart or chemically dismantled before burial rarely survive long enough to enter the geological record. Osedax interrupts that sequence in a remarkably effective way. After settling upon exposed bone, the worm begins dissolving the mineralized exterior through chemical secretion rather than physical force. Root tissues branch through the interior, producing expanding networks of channels that weaken the skeleton from within. As bacteria living inside those roots consume liberated collagen and lipids, the bone gradually loses both its internal architecture and much of the strength needed to resist collapse. The damage extends far beyond the small openings visible on the outer surface. Computed tomography imaging has shown that individual root systems merge beneath the exterior into extensive cavities spreading through the bone. What once appeared solid gradually becomes a delicate framework separated by interconnected tunnels. Eventually, the remaining structure may fragment under ordinary geological forces, leaving little capable of surviving the long journey toward fossilization. Those discoveries have reshaped how paleontologists interpret gaps within the marine fossil record. A missing skeleton no longer carries only one possible explanation. For generations, the absence of fossils from certain marine reptile groups was often interpreted as evidence that those animals were uncommon, geographically restricted, or poorly represented within ancient ecosystems. Osedax introduces another possibility. Some animals may simply have vanished before preservation ever became possible. Researchers now consider it plausible that a meaningful proportion of missing plesiosaur, mosasaur, and ichthyosaur specimens were never absent from prehistoric oceans at all. Instead, many carcasses may have settled onto seafloors where bone-eating worms had already established thriving populations. Colonization would have dismantled the skeletons long before sufficient sediment accumulated to protect them, removing evidence that later generations would otherwise have recovered. That realization changes the way paleontologists evaluate absence itself. The fossil record has always represented only a tiny fraction of organisms that once lived, yet Osedax demonstrates that biological destruction can systematically remove evidence before geology has an opportunity to preserve it. Some evolutionary lineages may therefore appear rarer than they truly were because their remains repeatedly entered environments where preservation was unlikely from the very beginning. The same process reaches into the history of whales. Once archaic whales began inhabiting ancient oceans during the Eocene, their skeletons also became available to Osedax. Evidence indicates the worms colonized those early whale bones just as readily as they colonize modern ones. Consequently, portions of the fossil record documenting early whale evolution were likely consumed before mineral replacement could begin, leaving additional gaps within an already incomplete history. Across approximately 100 million years, the influence of Osedax has extended beyond ecology into the preservation of life's own archive. Every dissolved skeleton altered the evidence available to future discovery. Every tunnel carved through ancient bone reduced the likelihood that another fossil would endure. In shaping the fate of bones on the seafloor, this unassuming worm has also helped shape humanity's understanding of prehistoric oceans, not by creating new records, but by determining which stories never reached the stone at all. Few living animals carry a history that stretches across the rise and disappearance of entire worlds. Long before the familiar giants of the modern ocean appeared, Osedax had already become part of the deep sea, and through each immense transition it remained. The same biological traits that allowed it to persist across ancient upheavals have quietly shaped its survival for roughly 100 million years, carrying the lineage through catastrophes that erased countless others. The greatest known turning point arrived at the end of the Cretaceous period, approximately 66 million years ago. During that mass extinction, roughly 75% of all species on Earth disappeared. Every non-avian dinosaur vanished, and the great marine reptiles that had dominated ancient seas, including plesiosaurs and mosasaurs, disappeared alongside them. For an animal whose fossil record now reveals a long association with those immense skeletons, the event represented the sudden loss of its principal source of nourishment. Yet, Osedax endured because its biology had never been tied to a single lineage alone. Although whales eventually became its best-known hosts, the worm possesses a far broader appetite than early researchers imagined. Modern observations and experimental evidence show that it can colonize the bones of many different vertebrates, including large fish, sea turtles, birds, and terrestrial mammals carried into the ocean. Its bacterial partners process collagen and lipid released from dissolved bone regardless of whether those nutrients originated inside a whale or another vertebrate. That flexibility transformed a seemingly specialized animal into a remarkably adaptable survivor. Sea turtles became especially important after the extinction that ended the age of marine reptiles. They survived the catastrophe and continued diversifying throughout the following millions of years. Their skeletons likely provided dependable opportunities for Osedax populations during the immense interval before the evolution of large whales. Rather than waiting for an entirely new ecological niche to appear, the worms continued exploiting bones already reaching the seafloor, preserving an evolutionary lineage across nearly 20 million years, until archaic whales eventually entered the oceans. Geography provided another layer of resilience. Fossil evidence indicates that by the Campanian age, Osedax already occupied a broad Atlantic distribution. Populations existed across widely separated marine environments rather than remaining confined to a single coastline or isolated basin. Such breadth greatly reduced the likelihood that one regional catastrophe could eliminate the entire lineage. Even when environmental conditions changed dramatically in one location, distant populations could continue surviving under different circumstances. Movement between those populations depended upon another enduring advantage. Adult Osedax remain anchored within bone for life, but their larvae drift freely through ocean currents before settling. That dispersal strategy allows recolonization whenever suitable habitat appears after local populations decline or disappear. As long as surviving larvae encounter fresh skeletal material somewhere within the network of currents, the lineage can expand once again into newly available territory. Recovery does not require every colony to survive. It requires only that enough larvae continue finding places where new generations can take hold. A fourth strength lies within the chemistry of the worm itself. Osedax routinely creates an acidic environment while dissolving mineralized bone through its root tissues. Living within conditions partly generated by its own metabolism means the animal regularly experiences chemical circumstances that would challenge many other deep-sea organisms. Although this does not make the worm immune to environmental change, it demonstrates an existing tolerance for unusual chemical conditions developed over an immense evolutionary history. Together, these traits formed a durable strategy: flexible feeding, broad geographic distribution, far-reaching larval dispersal. and resilience within chemically demanding surroundings carried Osedax through at least one confirmed mass extinction and possibly several additional periods of global environmental upheaval during its ancient history. The lineage survived, not because any single adaptation proved perfect, but because each strength reinforced the others. Across 100 million years, that combination repeatedly transformed disruption into continuity, allowing an obscure bone-eating worm to persist while entire dynasties of larger animals disappeared around it. By the time researchers finally recognized Osedax as a distinct branch of life, the worms had already accomplished something remarkable without attracting human attention. They were not confined to one isolated trench or a single stretch of continental slope. Instead, they had quietly established themselves across the world's oceans, occupying an immense vertical range that revealed an adaptability few scientists had imagined during the earliest years following their discovery. Initial observations after the discovery in 2002 suggested a straightforward picture. Every known specimen had come from deep water, leading many investigators to assume that Osedax belonged exclusively to the abyss. Depth appeared to define its existence, and the prevailing expectation held that these worms required environments deeper than roughly 3,000 feet, where whale falls were believed to persist undisturbed for long periods. That assumption gradually dissolved as additional surveys expanded beyond familiar research sites. One unexpected discovery came from the North Sea, where Osedax was documented at a depth of only 32 feet. Rather than inhabiting perpetual darkness alone, at least one species demonstrated that suitable bone, rather than extreme depth itself, determined where the animal could establish a colony. The finding transformed scientific understanding almost overnight, replacing a narrow ecological portrait with one far broader and more dynamic. As expeditions continued into different basins, the map expanded across nearly every direction. Confirmed records accumulated from the Pacific Ocean, the North Atlantic Ocean, the South Atlantic Ocean, the Southern Ocean, Antarctic waters, and even the Mediterranean Sea. Each new location demonstrated that the genus had spread across an extraordinary range of climates and oceanographic conditions, from comparatively shallow coastal environments to remote abyssal plains. Osedax repeatedly appeared wherever vertebrate bones settled onto the seafloor and remained available long enough for colonization. The deepest confirmed record arrived in 2013, when the human-occupied research vehicle Shinkai 6500 recovered Osedax brasiliensis from approximately 13,793 feet beneath the surface at the base of the Sao Paulo Ridge in the Day No. 1 South Atlantic Ocean. That discovery extended the known limits of the genus farther into the deep than ever before, confirming that these worms remained active under pressures reaching many hundreds of times greater than those experienced at sea level. Depth itself also shapes which species arrive. Long-term whale fall experiments conducted in Barkley Canyon revealed that different Osedax species preferred different levels within the water column, even when colonizing the same skeleton. A whale resting at one depth supported a community distinct from one positioned higher or lower along the canyon. Rather than forming a single uniform population, the genus organized itself into layered ecological neighborhoods, each adapted to particular environmental conditions. The bacterial partners living inside the worms reveal another dimension of this global pattern. Although every Osedax depends upon symbiotic bacteria within its root tissues, those microbial communities vary according to geographic region instead of the identity of the host worm alone. Populations inhabiting the Pacific Ocean carry bacterial companions that differ from those associated with relatives living in Antarctic waters, reflecting local evolutionary histories while preserving the same remarkable partnership. Modern observations also confirm that whales are only one chapter in the dietary story of this lineage. Living Osedax species have been documented consuming the bones of fish, sea turtles, marine birds, crocodiles, and terrestrial mammals carried into the sea. The flexibility first hinted at by the fossil record continues throughout the modern genus, allowing these worms to exploit vertebrate remains wherever suitable opportunities arise. At present, scientists have confirmed 32 species of Osedax. Even that total is widely regarded as incomplete. Only a minute fraction of the deep ocean has been explored in detail, and each expedition continues to reveal unfamiliar habitats, unexpected hosts, and previously unseen forms. The known distribution of Osedax, therefore, represents less a finished inventory than the visible outline of a lineage whose full diversity still lies hidden beneath the sea. The first signs of renewal appeared not in laboratories or policy documents, but on the ocean floor itself. Years after the greatest waves of industrial whaling had passed into history, fresh skeletons once again began settling into the darkness with increasing regularity. Each returning carcass became more than the remains of a single whale. It restored another link in a chain that had stretched across the oceans for millions of years, allowing distant communities to reconnect through the drifting lives of larvae carried by the currents. A decisive turning point arrived when the International Whaling Commission placed a moratorium on commercial whaling that took effect in 1986. The agreement did not erase the losses of the previous century, nor did it rebuild populations overnight. Even so, it marked the beginning of a gradual change. In several parts of the world, whales that had once become uncommon slowly increased in number, and with every additional generation, the probability grew that another immense skeleton would eventually descend to the seabed. Recovery unfolded unevenly. Humpback whales in some regions rebounded to levels approaching those believed to exist before industrial whaling transformed the seas. Their migrations became more familiar once again, carrying life across enormous distances before eventually returning nutrients to the deep. Other species have followed a far slower path. Blue whales in the Southern Ocean remain only a fraction of their historical abundance, despite decades of protection. The North Atlantic right whale continues to face severe decline, its future shaped by hazards that extend far beyond the legacy of whaling alone. Those differences are reflected beneath the waves. Regions where whale populations recovered more successfully began receiving new whale falls at intervals that more closely resembled the ancient rhythm of the ocean. Every fresh skeleton created another opportunity for Osedax larvae, for specialized invertebrates, and for the intricate succession of organisms that depends upon a complete whale fall. Another stepping stone appeared within the vast underwater network, narrowing distances that had once become too great for many populations to bridge. Marine biologists, monitoring recovering whale populations, also began documenting the return of specialist species to newly available habitat. Their appearance suggested that at least some members of these remarkable communities had endured decades of fragmentation, surviving in scattered refuges until additional whale falls allowed their descendants to disperse once more. The process unfolded gradually, measured over many years rather than seasons, reflecting the slow pace at which deep ocean ecosystems respond to change. Yet the rebuilding remained incomplete. Whale populations did not recover equally across every ocean basin, and the distribution of whale falls continued to mirror those uneven patterns. Some stretches of the deep sea regained a growing sequence of productive islands, while others remained separated by distances that challenged even larvae capable of drifting for hundreds of miles. The ancient stepping stone network therefore returned as an irregular mosaic rather than a continuous bridge. One of the clearest examples emerged in the Northeast Pacific, where many of the world's best-studied whale falls have provided an unparalleled window into deep ocean ecology. There, researchers observed encouraging evidence that recovering whale populations were restoring opportunities for organisms dependent upon these rare habitats. The pattern offered genuine optimism without suggesting that the system had fully healed, because important gaps remained between recovering populations and regions where whale numbers continued to lag behind historical abundance. Even as this gradual restoration unfolded, another transformation was beginning within the same waters. It did not arrive with harpoons or fleets, and it left no obvious scars upon the surface. Instead, it spread through changing ocean chemistry. During the early years of the 21st century, scientists working in parts of the Northeast Pacific detected a persistent decline in dissolved oxygen. The returning whale falls remained. Osedax continued its patient work, and the stepping stones slowly reappeared. Yet beneath that hopeful pattern, another pressure was expanding through the deep, altering the very water surrounding the communities that had only just begun to reconnect. Across the broad shelf beyond the coast of British Columbia, a patient experiment settled onto the seafloor with no expectation of spectacle. Its purpose rested in time itself, allowing an ordinary ecological process to unfold without interruption. If the patterns observed across other whale falls continued here, the sequence would reveal itself naturally. Instead, the passing years accumulated into evidence of an entirely different kind, one defined not by what appeared, but by what never did. Beginning in the early years of the previous decade, senior staff scientist Fabio De Leo of Ocean Networks Canada, serving also as an adjunct professor at the University of Victoria, helped establish an unusually ambitious study within Barkley Canyon. Humpback whale bones were carefully placed upon the seafloor approximately 62 miles southwest of Tofino, British Columbia, at a depth of nearly 3,000 feet. Rather than relying upon occasional expeditions separated by months or years, the site became part of a permanently connected underwater observatory, where the changing landscape could be followed without interruption as seasons and years drifted past overhead. The observatory represented one of the most sophisticated, long-term deep-sea research systems ever constructed. High-resolution underwater cameras remained fixed upon the bones, transmitting an uninterrupted visual record through Ocean Networks, Canada's cabled infrastructure. Every passing scavenger, every settling particle, every gradual alteration of the remains entered a continuous archive extending across an entire decade. Few ecological studies have been granted such remarkable continuity, removing much of the uncertainty that normally accompanies observations made only during brief research cruises. The expectations seemed well supported by earlier discoveries from multiple regions of the world. Wherever comparable whale bones had reached suitable depths, Osedax typically arrived within weeks or months. The drifting larvae required only an opportunity to encounter exposed bone before beginning the familiar transformation that had shaped deep-sea ecosystems for millions of years. Given sufficient time, colonization appeared less like a possibility than a dependable stage within the larger sequence of a whale fall. Year followed year, yet the expected arrival never occurred. The cameras continued their watch through changing conditions and recurring seasons beneath the surface. After 10 full years of uninterrupted observation, the record remained unchanged. No Osedax colonies established themselves upon the bones. No delicate crowns emerged from the skeleton. No branching root tissues entered the interior. Across the complete duration of the study, colonization remained entirely absent. The findings carried another layer that deepened their significance. Researchers also documented unusually low numbers of wood-boring bivalves belonging to the genus Xylophaga, another important ecosystem engineer responsible for breaking down submerged wood reaching the deep seafloor. Two unrelated groups of specialized recyclers, each depending upon different forms of organic remains, displayed the same unusual scarcity within the same environment. The pattern suggested a broader environmental pressure rather than the disappearance of only one organism. These results were published during 2024 in the journal Frontiers in Marine Science. Fabio De Leo summarized the implication with remarkable restraint, stating, "Basically, we're talking about potential species loss. Those few words reflected years of observation rather than momentary surprise. They arose from a data set that had patiently excluded alternative explanations through persistence instead of speed. Scientific discovery often celebrates unexpected appearances, yet carefully documented absences possess equal importance. When a process expected under established ecological understanding consistently fails to occur despite prolonged observation, that failure becomes evidence in its own right. The Barkley Canyon project, therefore, did not represent an unsuccessful investigation. It accomplished precisely what rigorous science seeks, an honest record of reality. Over 10 years, the observatory measured not the arrival of a hidden engineer, but the enduring presence of its absence, and that absence became the discovery itself. Long before researchers began searching for missing worms or measuring unexpected absences, the surrounding water had already begun changing in ways that could not be seen with the human eye. Bones could rest undisturbed upon the seafloor. Currents could continue their familiar paths, and the landscape might appear unchanged. Yet one of the ocean's most essential ingredients was gradually becoming less abundant. The transformation unfolded through chemistry rather than motion, reshaping the conditions that determined which forms of life could continue to exist. Oceanographers describe some regions of the sea as oxygen minimum zones, naturally occurring layers where dissolved oxygen falls to exceptionally low concentrations. Operationally, these zones are defined by oxygen concentrations between 0 and 20 micromoles per liter. They are not lifeless places, nor are they recent creations. Many have existed for immense spans of geological time, supporting communities specially adapted to conditions that would challenge countless other organisms. Their existence reflects the complex balance between circulation, biological activity, and the movement of oxygen through the ocean. Today, these oxygen minimum zones occupy between 1% and 7% of the total volume of the global ocean. That proportion remains relatively small, yet measurements collected across multiple ocean basins reveal a consistent pattern. The boundaries of these depleted layers are expanding, gradually reaching waters that previously contained enough dissolved oxygen to support a broader range of deep-sea life. The physical process behind this expansion is remarkably straightforward. Warmer seawater cannot retain as much dissolved oxygen as colder water. At the same time, increasing thermal stratification strengthens the separation between warmer surface waters and colder layers below. As this layering becomes more stable, oxygen-rich surface water mixes downward less efficiently. Deep water continues consuming oxygen through natural biological processes while receiving less replacement from above. Over years and decades, the deficit slowly grows, Barkley Canyon occupies a location already influenced by one of these naturally oxygen-poor regions. Even before recent warming trends intensified, conditions there rested close to the environmental limits tolerated by many deep-sea organisms. The canyon therefore existed near an ecological threshold, where relatively modest chemical changes could alter which species remained capable of surviving and reproducing. As oxygen minimum zones expand, that threshold does not remain fixed. The band of suitable habitat gradually shifts upward through the water column, reducing the amount of seafloor capable of supporting organisms dependent upon higher oxygen availability. For Osedax, this represents more than a change in preferred depth. It removes portions of habitat that once formed part of the connected network linking whale falls across the continental margin. The worm itself depends upon adequate dissolved oxygen, and so do the symbiotic bacteria living within its root tissues. Together, they form a partnership in which each participant relies upon the metabolic activity of the other. 2. When oxygen becomes severely limited, both members of that relationship experience physiological stress. Bone remains available, yet the biological machinery required to transform it can no longer operate with the same effectiveness. Evidence from Barkley Canyon indicates that this pressure extends well beyond a single genus. Wood-boring bivalves belonging to the genus Xylophaga, specialists that recycle submerged timber reaching the seafloor, have also declined within the same region. Their food source differs entirely from that of Osedax, but both occupy the same changing chemical environment. The pattern points toward a systemic driver rather than a predator, pathogen, or disturbance targeting one particular species. The Northeast Pacific margin, where Barkley Canyon lies, is regarded as one of the most oxygen-sensitive marine regions on Earth. Climate projections consistently indicate that continued warming will promote further expansion of oxygen minimum zones across major ocean basins. The transformation advances gradually, measured in changing concentrations rather than dramatic events, yet its influence reaches every organism whose survival depends upon the unseen chemistry carried within the water itself. Perspective changes when the scale of the sea floor comes into view. A single canyon, one carefully monitored experiment, and one unexpected absence can seem isolated until they are placed against the immense expanse beneath the oceans. The question is no longer confined to one location. It becomes a matter of how much of the deep world remains beyond observation, continuing its own history without witnesses. Everything deeper than approximately 3,280 feet forms the largest continuous environment on Earth. Together, these depths cover roughly 65% of the planet's entire surface. Across that immense landscape lie countless plains, ridges, canyons, trenches, seamounts, and sediment fields separated by distances measured across entire ocean basins. Each possesses its own currents, chemistry, and ecological rhythms, yet only a minute fraction has ever been examined in detail. Despite remarkable advances in ocean technology, the number of long-term, continuously instrumented deep-sea observatories remains remarkably small. Barkley Canyon stands among the most comprehensively equipped sites, supported by permanent cables, sustained electrical power, continuous communications, and an array of scientific instruments capable of recording changes over many consecutive years. Comparable systems exist at only a handful of locations worldwide, leaving nearly all of the deep ocean without uninterrupted observation. Even more striking is how little of the seafloor has ever been directly seen. Most of the deep ocean floor has never been imaged by any instrument at any meaningful resolution. Vast regions remain known only through broad mapping techniques that reveal the shape of the bottom while providing no view of the communities living upon it. Entire ecosystems continue their existence beyond the reach of cameras, sampling devices, or regular scientific visits. The discovery at Barkley Canyon required an extraordinary combination of effort and patience. Researchers intentionally transported humpback whale bones to the seafloor and positioned them within a carefully selected study site. Continuous camera surveillance followed for 10 uninterrupted years. Every frame required storage, processing, review, and scientific interpretation. Behind the images stood engineers maintaining the observatory, technicians operating complex equipment, and researchers capable of recognizing ecological change unfolding over a decade rather than during a single expedition. Few places possess the resources needed to sustain such an undertaking. Without that elaborate network, nothing unusual would have been recorded. Whale bones, resting on an unmonitored seafloor, would simply remain beyond human awareness. If Osedax failed to arrive, there would be no continuous record revealing its absence. The expected sequence would exist only as an assumption, while the missing stage would disappear into the surrounding darkness without leaving any obvious trace. This creates a difficult scientific challenge. Researchers currently possess no systematic method for determining where Osedax continues to thrive and where it may already have disappeared across oxygen-affected regions of the global ocean. Individual whale falls occur unpredictably, often in locations never revisited after discovery. Establishing identical long-term experiments across enough sites to measure broad ecological trends would demand resources extending far beyond what presently exists. For that reason, the Barkley Canyon observation cannot yet be confidently described as either exceptional or widespread. It may represent an unusual local circumstance confined to one canyon. It may also reflect a broader regional pattern unfolding simultaneously across numerous unmonitored whale falls separated by hundreds of miles. Present evidence cannot distinguish between those possibilities because the necessary observations simply do not exist. The deep ocean rarely offers immediate notice when its ecosystems begin changing. Transformations accumulate beneath miles of water, concealed from ordinary experience and often remaining undetected until an uncommon experiment, maintained with unusual persistence, reveals that something expected has quietly failed to happen. What eventually reaches the scientific record is therefore almost always delayed, inevitably incomplete, and perhaps only a small glimpse of changes already spreading across one of the largest habitats our planet has ever known. A whale fall is often described as a sequence, yet the word carries greater meaning than a simple list of stages. Each transformation depends upon the one before it, with no opportunity to skip a missing step. When one participant disappears, the remainder of the process does not simply slow. The entire ecological progression changes direction, leaving behind a very different landscape from the millions of years of evolution assembled. The missing stage begins with the absence already documented at Barkley Canyon. Osedax never arrives. The bones remain outwardly recognizable, resting upon the seafloor much as they first appeared after larger scavengers completed their work. Beneath that unchanged exterior, however, the chemical pathway expected to unfold has stalled before it can truly begin. The great stores of lipids sealed within the whale's skeleton remain locked inside mineralized bone. Under ordinary conditions, the worm's branching root tissues would penetrate deep into that reserve, allowing symbiotic bacteria to help unlock compounds that eventually nourish sulfate, reducing microorganisms living throughout the surrounding sediments. Their activity produces hydrogen sulfide, creating the chemical foundation upon which an entirely different community develops. Without the initial excavation performed by Osedax, that sequence is greatly diminished, and sulfide production remains suppressed. As hydrogen sulfide becomes scarce, the chemosynthetic organisms dependent upon it cannot establish themselves in the abundance normally associated with a mature whale fall. Microbial mats fail to expand into their expected extent. Tube worms, clams, mussels, and numerous other inhabitants adapted to this chemically rich environment encounter a habitat that never reaches the conditions required for successful colonization. The sulfophilic stage, ordinarily the longest and most biologically diverse chapter of a whale fall, never fully assembles. Within oxygen-stressed regions lacking Osedax, the skeleton itself becomes something unexpectedly static. Instead of serving as an engine driving decades of biological productivity, the whalebone functions largely as an inert mineral framework resting upon the sediment. It may persist for centuries with relatively little biological transformation, occupying space while supporting only a fraction of the ecological activity that would otherwise have surrounded it. The consequences extend beyond the bones themselves. The specialized whale fall organisms, found nowhere else on Earth, depend upon these productive habitats, appearing often enough to sustain connected populations. Every whale fall that fails to complete its ecological sequence represents another stepping stone that never becomes fully usable. For the confirmed specialist species restricted entirely to whale falls, each incomplete habitat reduces the opportunities for successful dispersal, reproduction, and long-term persistence. Another loss unfolds on a much smaller scale. Osedax does not merely consume bone. Its intricate network of bore holes and branching internal channels reshapes the skeleton into a remarkably complex surface filled with protected spaces and microscopic shelter. No other known organism produces this distinctive architecture. In its absence, the bone remains comparatively smooth, offering far fewer crevices where small invertebrates and microbial communities can establish themselves. Structural complexity, itself a valuable ecological resource, simply never appears. Carbon also follows a different path. Whale bones contain a concentrated reserve of organic carbon that ordinarily re-enters the surrounding food web through biological processing. When decomposition remains incomplete, much of that carbon stays locked within mineralized tissue rather than circulating through successive generations of deep-sea life. Over immense stretches of time and across countless whale falls, this altered pathway becomes part of the broader carbon budget of the deep ocean, where even gradual shifts can accumulate into measurable ecological consequences. Researchers emphasize that the complete magnitude of this cascading process remains an active subject of scientific modeling. Many details continue to be refined as new observations emerge from long-term studies. Yet one essential point no longer rests entirely within theory. The decade of uninterrupted monitoring at Barkley Canyon demonstrated that the first missing step can indeed occur, and once that opening link disappears, the remainder of the ecological chain no longer unfolds in the way evolution shaped it across millions of years. A chain can remain unbroken even after one of its links has vanished from view. For a time, the remaining connections continue carrying their weight, giving the appearance of continuity. Only later does the strain reach the neighboring links, spreading outward until the absence that once seemed local begins to reshape the entire structure. Across the deep ocean, the network created by whale falls follows much the same pattern, its strength resting not upon any single carcass, but upon the living connections carried between them. Every new generation of Osedax begins with adults already established inside whale bones. Mature females release eggs that develop into drifting larvae, each carried by currents through the surrounding water in search of another skeleton resting upon the seafloor. The larvae possess no reserve plan if that source disappears. Without reproducing adults, there are no eggs. Without eggs, there are no larvae. Without larvae, the next whale fall remains only a collection of untouched bones. As expanding oxygen-depleted waters render formerly suitable depths uninhabitable, the first loss is not the larvae, but the adults anchored within existing whale falls. Once those established populations disappear, reproduction stops across that affected region. Future whale carcasses continue sinking exactly as before, arriving with their stores of bone lipids intact, yet the surrounding water contains no local generation of Osedax prepared to discover them. The expected colonizer never arrives because it no longer exists within dispersal distance. The result transforms each whale fall into an isolated destination rather than a connected waypoint. Bones remain available, but availability alone cannot sustain a species that depends entirely upon successful arrival. Habitat without colonists becomes ecologically unreachable, much like an island surrounded by an ocean no traveler can cross. Fabio de Leo described this consequence in remarkably direct language while reflecting upon the observations from Barkley Canyon. The connectivity formed by these island habitats, he explained, will no longer exist, allowing diversity among regional populations of Osedax to begin disappearing across broad stretches of the ocean. His words describe more than the loss of a single worm. They describe the gradual unraveling of an ecological network assembled over millions of years through repeated cycles of dispersal and colonization. Isolation rarely remains confined to one generation. Neighboring populations that once exchanged drifting larvae become increasingly separated as intervening regions lose suitable habitat. Genetic exchange declines because fewer individuals successfully travel between surviving populations. Diversity gradually narrows, reducing the range of inherited traits available for adapting to future environmental change. Each disconnected population becomes more dependent upon its own limited genetic variation, making long-term persistence increasingly uncertain. The same fragmentation extends beyond Osedax itself. The confirmed whale fall specialist species that depend upon productive sulfophilic communities also rely upon connected habitats for successful dispersal. Their larvae drift through ocean currents, searching for another suitable whale fall before their brief developmental window closes. When successive stepping stones fail to develop into functioning ecosystems, those journeys end without settlement. Regional populations become divided into smaller groups, separated by stretches of unsuitable habitat that continue growing wider through time. Unlike many ecological forecasts, this sequence does not rest solely upon theoretical modeling. The first stage has already been documented through prolonged observation. For at least 10 years, the experimental whale bones resting within Barkley Canyon remained without Osedax colonization despite continuous monitoring. Throughout that decade, no local adults established themselves, no local larvae emerged from those bones, and no new contribution entered the surrounding dispersal network. Across the broader ocean, many connected pathways almost certainly continue functioning, as they always have. Yet wherever these living links disappear, the network loses more than individual organisms. It loses movement, exchange, inheritance, and continuity, each fading one region at a time, until an ocean that still contains whale falls gradually becomes less connected than the one that existed before. The longest surviving lineages often carry an appearance of permanence. Across immense spans of geological time, they endure changing continents, shifting oceans, vanished predators, and worlds transformed beyond recognition. Their continued existence suggests resilience measured not in centuries, but in epochs. Yet the history of Osedax reveals an unexpected contrast. The challenge now emerging is unlike the catastrophes that shaped its past, not because it is more sudden, but because it advances with remarkable persistence. Across the past 100 million years, Earth experienced repeated biological crises that reshaped life on a planetary scale. The impact marking the end of the Cretaceous Paleogene extinction transformed ecosystems within years to decades by geological standards. Earlier intervals, marked by widespread oxygen depletion, unfolded across thousands of years, altering marine environments before gradually giving way to more stable conditions. Each event carried extraordinary destruction, yet each eventually reached an end. Dust settled from the atmosphere, volcanic activity diminished, ocean chemistry slowly recovered, and surviving populations expanded once favorable habitats returned. That recurring pattern offered Osedax opportunities for renewal. Even after the disappearance of entire groups of marine reptiles, enough populations remained scattered across the oceans to rebuild their distribution. Their broad geographic range, flexible diet, drifting larvae, and remarkable tolerance for chemically demanding environments combined into a strategy that repeatedly outlasted upheaval. The crises were immense, but they were temporary. Recovery followed because the conditions preventing expansion eventually faded. The situation now unfolding follows a different rhythm. Oxygen minimum zones do not retreat naturally, while the underlying warming of the ocean continues. As seawater absorbs additional heat, its ability to retain dissolved oxygen steadily declines. Stronger thermal layering further limits the downward movement of oxygen-rich surface water into deeper environments. Instead of a disturbance that arrives and passes, the chemical landscape itself shifts toward a new equilibrium that continues moving in the same direction for as long as the forcing remains. For Osedax, retreat is not a simple option. The whale bones that sustain every stage of its existence descend into depth ranges determined by the natural sinking of massive carcasses. Those destinations increasingly overlap with expanding regions where dissolved oxygen becomes too scarce for healthy populations. Moving into shallower water would separate the worm from the resource upon which its entire life cycle depends. Remaining within the affected depths exposes both the worm and its bacterial partners to conditions that steadily undermine their survival. The mechanism that once restored damaged populations also encounters a fundamental obstacle. Larval dispersal carried Osedax back across emptied habitats after earlier catastrophes because adults remained somewhere within the surrounding network, continually releasing new generations into ocean currents. Where adult populations disappear entirely, that source of larvae disappears as well. Recolonization from within the affected region becomes impossible because the reproductive engine has already stopped. Recovery depends upon larvae arriving from surviving populations beyond the collapsed area, a possibility that becomes less likely as disconnected regions grow larger. Evolution provides no immediate rescue. The known diversity of Osedax represents millions of years of gradual divergence shaped by changing oceans and shifting ecological opportunities. The environmental pressure created by expanding oxygen minimum zones is unfolding across mere decades, a pace that offers little opportunity for entirely new adaptations to spread through regional populations before habitats become unsuitable. Taken individually, none of the characteristics that sustained Osedax through earlier mass extinctions has completely vanished. Generalist feeding still broadens its potential resources. Wide distribution still places populations across distant oceans. Larval dispersal still functions wherever adults remain established. Chemical resilience still allows survival under conditions many organisms cannot tolerate. Together, however, these strengths now confront a form of environmental change that weakens each one simultaneously. The paradox is striking. A lineage that endured planetary catastrophes for 100 million years now faces a challenge defined not by explosive destruction, but by the gradual movement of an invisible chemical boundary that does not simply pass, then allow the old balance to return. Knowledge of Osedax continues to grow through an unusual combination of paleontology, marine biology, and long-term ocean observation. Each discipline contributes a different piece of the story. Fossil collections preserve evidence from vanished oceans. Experimental whale falls reveal how living communities assemble today, and permanently installed observatories record changes that would otherwise pass unnoticed beneath thousands of feet of water. Together, these approaches are transforming a creature once entirely unknown into one of the best documented architects of the deep sea floor. Among the most valuable research tools is the cabled observatory operated by Ocean Networks Canada. Extending across the northeastern Pacific, this system provides one of the most extensive, continuous deep-sea monitoring networks ever established. Power and data flow constantly between the seafloor and the surface, allowing high-resolution cameras and scientific instruments to operate throughout the year without depending upon short research expeditions. The decade-long record from Barkley Canyon existed only because this infrastructure remained active day after day, preserving an uninterrupted history that no visiting expedition could have assembled. Elsewhere, researchers continue placing experimental whale bones at carefully selected depths under differing environmental conditions. Some sites lie within well-oxygenated waters, while others occupy regions where dissolved oxygen approaches the lower limits tolerated by deep-sea communities. By comparing how rapidly Osedax appears, how successfully colonies develop, and whether reproduction follows, scientists hope to determine exactly how strongly dissolved oxygen influences every stage of the worm's life cycle. Each new deployment becomes another carefully measured reference point against which future observations can be compared. Evidence is also emerging from specimens collected long before anyone had heard the name Osedax. Museum collections around the world contain marine reptile and early whale fossils gathered across generations of paleontological exploration. Increasingly, these bones are being examined with computed tomography scanning, allowing researchers to inspect their interiors without damaging the specimens themselves. Hidden tunnels, branching cavities, and distinctive bore patterns can now be identified inside fossils that appeared completely ordinary from the outside, extending the historical record of the worm farther into Earth's past. During the year 2025, researchers associated with the Natural History Museum in London published the most detailed examination yet of fossil Osedax traces. Studying bones belonging to plesiosaurs, mosasaurs, and ichthyosaurs, they identified 7 previously unrecognized ichnospecies preserved within the ancient borings. Rather than documenting the worm itself, these fossil structures reveal the diversity of behaviors left behind, providing the clearest picture so far of how varied Osedax communities had already become during the age of marine reptiles. Living populations are now being compared across contrasting environments as well. Scientists continue examining regions where Osedax remains abundant under healthy oxygen conditions, alongside locations influenced by expanding oxygen minimum zones. Establishing a reliable baseline for normal colonization rates has become increasingly important, because only with that reference can researchers measure how unusual the prolonged absence observed at Barkley Canyon truly is. Although the evolutionary history of Osedax reaches back approximately 100 million years, its scientific history remains remarkably brief. The genus has been known for only 24 years. In that short time, discoveries have progressed from identifying an unfamiliar worm upon whale bones to reconstructing its ancient fossil record and documenting signs that some modern populations may already be under environmental pressure. The field remains young, its methods continue advancing, and each new observation adds another carefully measured piece to a story that is still being written across both the living ocean and the rocks, preserving its distant past. Every whale carries within its skeleton far more than structural support. Bone is built primarily from calcium phosphate, together with smaller amounts of calcium carbonate, forming a durable framework that encloses collagen, lipids, and many other organic compounds accumulated throughout the animal's life. Long after soft tissues have disappeared, these buried reserves remain stored within the skeleton, creating a remarkably concentrated package of biological carbon resting upon the seafloor. In a functioning whale fall, that stored material does not remain untouched forever. Osedax and the bacterial partners living within its root tissues gradually unlock the organic compounds hidden inside the bone. Their work unfolds across years and sometimes decades, feeding a sequence of organisms that together recycle carbon back into the surrounding deep ocean ecosystem. The process is deliberate rather than rapid, yet it continually transfers material from an apparently lifeless skeleton into living communities that depend upon this rare source of nourishment. When Osedax is absent, that pathway changes fundamentally. The skeleton still reaches the seafloor carrying the same reserves of carbon, but the biological mechanism that normally begins extracting them never develops. Collagen remains enclosed within mineralized tissue. Lipids remain trapped deep inside dense bone, instead of passing through bacteria, invertebrates, and the wider food web. Much of that organic material stays embedded within the skeleton as the surrounding mineral framework slowly weathers through physical abrasion and inorganic chemical reactions unfolding across centuries. The contrast illustrates how differently time operates in the deep ocean. Biological recycling can reshape a whale skeleton within the span of decades when the complete community assembles. Without that community, the same bones may persist far longer, changing only through gradual processes that move at the pace of currents, sediments, and mineral chemistry. Carbon remains present in both situations, yet the route it follows through the environment becomes profoundly different. This distinction matters because the deep ocean already serves as Earth's greatest long-term reservoir for carbon. Organic material that sinks into deep water and escapes rapid biological recycling may remain isolated from the atmosphere for extraordinarily long intervals. Countless natural processes contribute to that immense storage system, from drifting marine snow to sediments accumulating across abyssal plains. Whale bones represent only one component of this broader cycle, yet they concentrate substantial amounts of organic material within a relatively compact structure. Scientists are still working to determine how much Osedax contributes to the global movement of carbon through these environments. The research field remains young, observations remain geographically limited, and many whale falls have never been monitored from beginning to end. Without broader records extending across multiple oceans and many decades, precise estimates remain beyond present knowledge. That uncertainty reflects the difficulty of measuring one biological pathway across the largest habitat on the planet, rather than any doubt that the pathway exists. What current research can establish is more focused and equally important. Where bone decomposition is interrupted because Osedax fails to colonize, the flow of organic carbon changes. Less material returns through biological recycling, while more remains locked within slowly degrading skeletal minerals. The direction of that shift is clear, even though its full magnitude has not yet been calculated. Over centuries, repeated changes of this kind have the potential to influence the chemistry of deep ocean environments in ways scientists are only beginning to examine. Viewed from the scale of a single worm, the transformation seems almost impossibly small. Viewed across generations of whales, thousands of whale falls, and millions of square miles of seafloor, the accumulation becomes far more difficult to overlook. A creature scarcely noticed until recent decades participates in one of the ocean's oldest conversations, guiding the slow return of carbon through living systems and reminding us that even the smallest participants can leave enduring marks upon the chemistry of an entire world. For a full decade, the evidence did not arrive as a dramatic discovery or an unexpected appearance. It arrived as the steady accumulation of ordinary days, each one adding another frame to a record that gradually became extraordinary through what it never revealed. Far below the rolling surface of the Northeast Pacific, cameras remained fixed beside a carefully placed set of humpback whale bones resting on the floor of Barkley Canyon. Connected by cables stretching across the seafloor, the observatory received continuous electrical power and transmitted images in real time to research stations on shore. Beyond the reach of sunlight, the camera's own illumination defined the scene. Each recording began with the same familiar view: pale bones resting against dark sediment, surrounded by water that never carried the changing light of day. The earliest recordings showed that the deep sea was anything but lifeless. Small scavengers approached the skeleton within the first weeks. Tiny invertebrates explored exposed surfaces. Sediment surrounding the bones shifted with the movements of burrowing animals, and traces of biological activity appeared exactly where researchers expected them. The experiment had succeeded in placing a whale fall into an active deep ocean environment. Life had arrived. Yet another arrival never came. Across published whale fall studies from many parts of the world, the sequence had become familiar. Within weeks or months, delicate red plumes would begin emerging from the bone surface as Osedax settled and established itself. Beneath those feathery crowns, living root structures would penetrate deep into the skeleton, unlocking the organic material hidden inside. The smooth surface of the bones would gradually transform into a crowded landscape, supporting an expanding community of specialized organisms. The Barkley Canyon recordings preserved none of that transformation. Each review of the footage returned to bones that looked remarkably unchanged. Weeks became months, months became years. Layer after layer of observations accumulated, while the expected colonization remained absent. No crimson crowns appeared beneath the camera lights. No intricate network of roots spread through the skeleton. The surfaces never developed. The textured complexity, documented at thriving whale, falls elsewhere. Instead, the bones remained largely exposed, visited by ordinary scavengers, yet untouched by the engineer that should have reshaped the habitat from within. For the researchers studying those recordings, the significance emerged gradually because they understood precisely what they were meant to find. They were not searching for an unknown phenomenon. They were watching for one of the most consistently observed stages of whale fall succession. Every passing month without Osedax increased the weight of the observation. Every additional year strengthened it further. By the conclusion of the decade, the result had become exceptionally clear. Continuous monitoring had removed many of the uncertainties that often complicate deep-sea research. The cameras had never stopped recording. The experiment had not depended upon isolated visits separated by years. The evidence existed as an uninterrupted visual history extending from the placement of the bones through 10 complete years of observation. In scientific research, a confirmed negative result carries the same evidentiary value as a confirmed positive one when the observations are sufficiently thorough. This experiment satisfied that standard. The absence was not inferred from missing samples or incomplete surveys. It was documented continuously by an instrument that remained present throughout the entire life of the study. Few deep ocean experiments achieve that level of persistence. The bones remain on the floor of Barkley Canyon, illuminated whenever the observatory turns its gaze toward them. They still mark the place where an ecosystem should have unfolded through familiar stages of succession. The structure that Osedax would have carved, the habitat it would have assembled, and the community it would have sustained were never recorded, because over 10 uninterrupted years, they were never there to record. History sometimes survives in forms so small that its scale becomes almost impossible to recognize. Resting within the bones of dead whales, hidden beneath miles of ocean water, Osedax carries a biological story that began long before humanity existed and has continued with remarkable consistency across worlds that scarcely resemble one another. More than 100 million years ago, when plesiosaurs still crossed ancient seas, these worms were already performing essentially the same task they perform today. Their root tissues released acid that gradually dissolved bone. Symbiotic bacteria transformed nutrients locked within the skeleton into usable energy. Females remained anchored inside the remains of enormous marine animals, while tiny males lived within their gelatinous tubes, their development shaped not by inherited destiny alone, but by the environment surrounding them. The details discovered through living specimens and fossil evidence align with remarkable precision across immense stretches of geological time. Those ancient oceans differed profoundly from those of the present. Continents occupied unfamiliar positions. Ocean circulation followed different pathways. Marine reptiles dominated ecosystems that vanished tens of millions of years before the first whale ever swam through salt water. Countless species appeared and disappeared, climates shifted repeatedly, and the chemistry of entire oceans changed through the rhythms of Earth's long history. Even within those changing worlds, Osedax continued its patient occupation of bones, descending toward the seafloor. When the asteroid impact marked the beginning of the Cretaceous-Paleogene extinction, the familiar architecture of marine ecosystems unraveled. Many dominant lineages vanished forever, yet somewhere beneath those altered seas, surviving populations of bone-eating worms endured. As the dust settled and oceans gradually stabilized, they remained part of the recovering deep sea, continuing a lineage that had already weathered one of the greatest biological upheavals the planet had ever experienced. Their story extended naturally into the Paleocene and the Eocene. During those distant ages, small hoofed mammals gradually adapted to life near water, giving rise over millions of years to the earliest whales. Those transitional animals eventually abandoned the shoreline entirely, becoming fully marine descendants unlike anything that had come before. As whale populations expanded across the oceans, They created an entirely new source of enormous skeletons descending into the abyss. Osedax did not invent a new strategy. It simply encountered a new abundance of familiar opportunity. For approximately 45 million years, whales and bone-eating worms have shared an ecological partnership separated by death rather than life. Every whale reaching the seafloor offered another habitat, another source of nourishment, another bridge connecting distant regions of the deep ocean. As whales diversified into countless forms, Osedax diversified alongside them. Today, scientists recognize 32 living species, each representing millions of years of evolutionary separation from a common ancestor while retaining the same essential way of living. Their descendants now inhabit every major ocean basin. They have been documented in protected Scandinavian fjords near Sweden, along continental margins, within the Pacific, the Atlantic, the Indian Ocean, and the Southern Ocean. Wherever large bones settle into suitable conditions, members of this ancient lineage possess the potential to arrive, establish themselves, and begin the gradual transformation that has defined their existence across geological time. Despite that extraordinary history, each individual remains astonishingly modest in scale. An adult female typically measures between 1 and 3 inches in length. She possesses no eyes to perceive her surroundings, no mouth to consume food directly, and no ability to leave the skeleton once established. Nearly her entire existence unfolds embedded within the remains of another creature, nourished through chemistry, bacteria, and bone beneath complete darkness. Across 100 million years, continents have wandered, oceans have reorganized, and dominant forms of marine life have risen and disappeared. Through every transformation that still allowed its habitat to exist, Osedax remained one enduring thread woven through the deep ocean, carrying an ancient continuity inside one small, hidden life. No organism keeps forever what it gathers during its lifetime. Across the ocean, every living thing participates in an exchange that began long before its birth and continues long after its death. A whale spends decades collecting energy and nutrients from countless meals spread across immense migrations, yet none of that abundance truly belongs to the whale alone. It is borrowed from the surrounding sea, held for a time within muscle, blood, blubber, and bone, then entrusted back to the world that provided it. When that journey ends, and the body descends through the water column, the return begins. The whale fall is not merely the resting place of a giant animal. It is the mechanism through which an enormous store of biological wealth is distributed with remarkable order. Nothing happens all at once. Every stage unfolds at its own pace, each one preparing the conditions required for the next community to emerge. Large scavengers arrive first, drawing nourishment from soft tissue that would otherwise remain inaccessible. Smaller animals continue the work after the larger visitors depart, reducing what remains with increasing precision. Microorganisms expand into newly exposed surfaces, reshaping the chemical landscape of the surrounding sediment. Time itself becomes part of the process, allowing one community to inherit the opportunities created by another. Eventually, the bones become the final great reserve. They appear rigid and permanent, yet within their mineral framework remain collagen, lipids, and other organic compounds accumulated throughout the whale's long life. These substances represent one last concentration of biological carbon and chemical energy waiting beneath a shell of calcium phosphate and calcium carbonate. Without access to that hidden store, the story of the whale fall remains unfinished. Osedax provides the missing step, Through delicate root tissues, the worm gradually opens pathways into the skeleton using carefully controlled acid secretion. Symbiotic bacteria living within those roots transform compounds locked inside the bone into nourishment that sustains the animal itself. As those reserves are released, conditions shift once again. Sulfide production increases, new chemical gradients emerge, and the final ecological community begins assembling around a resource that had remained inaccessible until the bone-eating worm arrived. The significance of that transformation extends far beyond a single skeleton. The last phase of a whale fall can endure for decades, supporting organisms found almost nowhere else. Species that specialize in these habitats depend upon the sequence remaining intact from beginning to end. Their existence rests not simply upon the arrival of a whale, but upon every preceding stage, successfully handing the ecosystem forward until the deepest reserves are finally opened. When Osedax never appears, the transfer pauses before completion. The bones persist, but much of what they contain remains locked within mineral structures that weather only through slow physical and inorganic chemical change. The final ecological community fails to assemble as expected, and the pathway through which nutrients once returned to the surrounding deep sea is interrupted. What disappears is larger than a single interaction between one worm and one skeleton. A process maintained across at least 100 million years no longer unfolds in its familiar form. Seen from that wider perspective, the ocean reveals itself as an immense network of transfers rather than isolated lives. Energy captured by microscopic algae passes into drifting zooplankton. Fish gather that energy into larger bodies. Whales concentrate it across decades of migration before carrying it toward the seafloor at the end of life. Osedax receives what remains within the bones, allowing countless other organisms to inherit resources that would otherwise remain inaccessible. Every transfer depends upon the one before it. Remove a connection that has endured across 100 million years, and the sequence does not merely lose a single participant. The pathway itself changes, carrying the history of the ocean in a different direction than the one it followed for ages beyond ordinary imagination. Some of the oldest systems on Earth have never depended upon being seen. They continue their work beyond the reach of sunlight, beyond familiar landscapes, beneath waters that conceal almost everything from ordinary experience. Among them is a creature with no mouth to feed, no eyes to observe its surroundings, and no ability to travel once it has settled into a home. For longer than mammals have existed, Osedax has remained part of a process that quietly shaped the deep ocean without ever asking to be noticed. Most people will live their entire lives without hearing its name. They may watch whales breach along distant coastlines, read about migrations spanning entire oceans, or imagine the immense blue world beneath a ship's hull, yet never learn that every great whale eventually becomes part of another story. Fewer still will discover that a whale fall unfolds across generations, or that one small worm has helped complete that transformation for at least 100 million years. That hidden continuity reflects the character of the deep sea itself. Far below the surface, immense ecological systems continue without applause or spectacle. Extreme pressure, perpetual darkness, and near-freezing water have never prevented life from constructing relationships of extraordinary complexity. Long before forests spread across familiar continents in their modern form, before birds crossed the skies, before humanity appeared, these unseen exchanges were already unfolding beneath the waves. Because they remain hidden, changes within them often pass unnoticed as well. Nothing at the ocean surface marks the disappearance of a bone-eating worm from a canyon thousands of feet below. No unusual color spreads across the water, no ripple reaches passing vessels. A whale may still swim overhead, seabirds may still circle above the waves, and coastal weather may continue unchanged. From every ordinary human perspective, the ocean appears exactly as it did before. Yet, beneath that familiar surface, the Barkley Canyon experiment recorded something profoundly different. For 10 complete years, cameras continued their patient observation beside a whale skeleton resting on the seafloor. The archive grew frame by frame, season after season, preserving an uninterrupted history of what occurred around those bones. The record remains available, not because it captured an astonishing arrival, but because it documented an astonishing absence with unusual certainty. The skeleton still occupies its place within the canyon. Smaller scavengers visited, sediment shifted, ordinary deep-sea activity continued around it. The transformation expected by decades of whale fall research never unfolded. The crimson feeding plumes that should have extended into the surrounding water never appeared. The intricate root structures that should have entered the skeleton never developed. A community expected to emerge simply never assembled. Archives preserve many remarkable discoveries, yet this collection of images tells its story through repetition rather than surprise. One recording resembles the next with remarkable fidelity. Another month passes. Another year follows. The bones remain largely unchanged. Scientific certainty accumulates not through a dramatic moment, but through the persistence of observation extending across an entire decade. That pattern carries a broader lesson about the ocean itself. The deep sea rarely announces what it is gaining or losing. Its transformations usually occur beyond direct human awareness, recorded only when instruments happen to be present for long enough to recognize that something expected has failed to happen. Countless other places remain unobserved, their histories unwritten, except by the organisms living there. Perhaps that is the deepest character of this world beneath the waves. It continues its ancient work with remarkable patience, carrying forward relationships established across geological ages, while revealing only fragments to those watching from above. When one of those ancient threads begins to fray, the ocean does not proclaim the change. It simply becomes a little less complete, one hidden place at a time, with almost no witnesses except the creatures that remain. The thread does not begin with a whale. It begins far deeper in time, when a plesiosaur drifted through an ancient sea and settled onto a seafloor unlike any living ocean. Long before familiar coastlines, before grasses covered continents, before mammals came to dominate the land, its skeleton became part of a sequence that had already found its rhythm. A small worm reached the bones, dissolved mineral with acids released from living tissue, partnered with bacteria hidden within its roots, and opened a path for life that would continue long after the great reptile itself had vanished. The remains were not abandoned, they were transformed. That same sequence endured through changes almost impossible to imagine. Continents shifted their positions, ocean currents reorganized, entire groups of marine animals appeared, flourished, and disappeared. The Cretaceous-Paleogene extinction reshaped life across the planet, yet the lineage, represented by Osedax, persisted. Fossil evidence shows it continued into the Paleocene and the Eocene, eventually encountering a new source of bone as the earliest whales returned to the sea. Over millions of years, whales diversified, and the worms diversified beside them, forming new species adapted to different depths, temperatures, and hosts, while preserving the same remarkable strategy for unlocking nutrients sealed inside bone. Today, that process continues across much of the world ocean. Whenever a whale settles onto a suitable seafloor and oxygen conditions remain favorable, a familiar progression can unfold. Scavengers arrive, smaller organisms follow, bones become the foundation for another community, and the last stores of organic material gradually return to surrounding ecosystems. The exchange is unhurried, extending across years and often decades, linking one generation of deep-sea life to the next through a chain that has operated for an extraordinary span of geological history. Yet one carefully observed place tells a different story. At Barkley Canyon, the expected arrival never came. For more than 10 years of continuous monitoring, researchers watched whale bones remain without the flourishing Osedax community documented at many other whale falls. That observation does not demonstrate the fate of every deep ocean basin, nor does it reveal the future of the species as a whole. It does show, with unusual clarity, that an interruption can occur under present-day conditions, and that such an interruption has consequences extending beyond the absence of a single animal. Across many regions of the ocean, warming waters are associated with the expansion of oxygen minimum zones, areas where dissolved oxygen becomes scarce enough to challenge many forms of life. Scientists are actively investigating whether those changing conditions help explain why Osedax failed to establish itself at Barkley Canyon, and whether similar patterns are emerging elsewhere. Those questions remain the subject of ongoing research, supported by long-term observatories, experimental whale falls, museum collections, and years of painstaking analysis. The answers are still unfolding. Whatever those answers ultimately become, the story of Osedax offers a different way of seeing the living planet. It reminds us that enduring systems are often built from organisms that attract almost no attention, performing work that rarely reaches human awareness. A worm only a few inches long, lacking eyes, lacking a mouth, and rooted inside bone, has participated in one of the ocean's longest ecological relationships. Its history reaches backward through vanished seas and extinct giants, connecting worlds separated by 100 million years. The next chapter of that history has not yet been written. It will emerge from careful observation, patient evidence, and the changing conditions of the ocean itself. For now, the bones remain where they settled, the cameras continue their watch, and the thread that stretches from an ancient plesiosaur to a modern whale still reaches across time, inviting us to understand how even the smallest lives can become part of Earth's longest stories.